Welcome, grade 12s. Welcome to our show. We're not chatting about the skeleton today because we were talking about it in grade 11.
We couldn't move this because Indy is absolutely besotted.
I'm in love. This
is his name?
Dudu.
Yeah.
Zile. This is my friend. So guys, grade 12s, let's get to your show, enough with grade 11 because they've had their turn. So, we are talking about meiosis. And this is going to be an awesome, awesome, awesome lesson. Kathy has been teaching straight for 2 hours and she's doing fantastic. You know what, guys? Chat to me as well. Let me know.
Please ask questions.
Yes,
okay.
Guys, where
it's
are
so
you?
that I want to know that you're watching and that you're enjoying it and that you're learning. Okay, because if you ask a stupid question, then oops, I've got to go and teach it again. And if you ask a really intelligent question, I think, whoa, I need to tackle that one.
Let's impress Kathy. That's what my aim for today is. Guys, I want impressive questions. I want Kathy to say, wow, that's a good question.
This from someone who's in love with the skeleton. I shudder to think. I mean, can you imagine?
I'm not the one asking questions.
If we had done this yesterday, you could have had a Valentine.
I could have.
Yes.
Well, maybe I'll redo my Valentine. Well, Dudu and I are going to be behind the computer screen here. We're going to go to an ad break, and we'll see you in a second. Bye. Hi guys, and welcome back. Today is going to be a cracking show and I'm so excited. Anele has been tuning in since 4 o'clock. Kathy, she's been with you since grade 10. She's a matric, but she's tuning in anyway. Good. Anele, you are a mind setter of notes. Let's get straight to Kathy.
Perfect, hun, because you know what? You have to know about blood and anemia and blah, blah, blah. And you know what? You understand it now because I've reminded you from your grade 10 work. So it's really, all you're doing is you're revising. And every single little bit helps. And I mean, if you're going to be sitting on the couch, instead of watching the 50th retake and reversion of something like Generaties, I'm not supposed to say the word, okay? This is far better, don't you think? Perfect. So Zanella, thank you very much, darling. And people, I forgot just now with the grade 11 session, to say a very big thank you to John and the butcher for organizing our bones, and a huge thank you to St. Stithians Girls College, who actually lent us their Giles. So, and we're going to be borrowing him again next week. So there we go. Thank you very much, ladies. We really appreciate you sharing your boyki with us. Alrighty, we're going on to meiosis. Now, people, you remember we had mitosis. And you know what? They are quite capable of asking you for a comparison between meiosis and mitosis. Mitosis was for growth, for repair, for replacement, and for reproduction in unicellular organisms. Okay, now let's think about this. Mitosis, growth, your cells have to grow. Your hair grows, your nails grow, you grow in size, you grow in weight, you decrease in weight, you grow in weight. People that do gym, guys that train. My son at the moment is going nuts for gym training and fitness training for rugby. What's happening? The body has to make new muscle cells. Why? Because it says, oh my gosh, I can't cope with the 10 I've got. I'm going to have to make 15 or 20 or 100 or 1,000 or whatever the case is. Okay, so for growth, we have to fix things. We have to repair things when they break. So when cells break, we must replace them. Now, can you imagine that if you have maybe a very dry lip and your lip cracks, and instead of lip cells growing there, you end up with heart cells pumping away, so cardiac cells, or you end up with liver cells growing and just being made in place of your lips. No. Why? Because with mitosis, we start off with a diploid cell. Okay? It's a complete cell. It's got both pairs of chromosomes from mom, from dad. It is a perfect normal body cell. Okay? Any body cell. It's called the somatic cell. And the diploid cell then literally duplicates. And when it duplicates, it makes an identical copy of itself. So we have an absolute, it's like a photostat machine. All right? So my lip cells, well, which lip cells are going to repair that hole? The other lip cells on the sides. If there's a heart cell that dies, it's the heart cell next to it that's going to replace it. Make a new one. So you've always got identical cells, always. So we start off with a diploid cell, and we end up with two identical. They are identical in every possible way. Identical daughter cells. Okay, perfect. Absolute duplicates. So we're growing, we're repairing, we are replacing cells. So if a cell gets damaged and hurt and it can't be repaired, well, break it down into its basic components, which is carbohydrates, fats, lipids, and yay, goodbye, mwah, have a wonderful day, clean out, sweep everything away and make a new one. So we have replacement. And then reproduction, but people look at this, unicellular. A unicellular organism is one cell. And if it's only one cell, okay, one cell, there aren't any other cells around to help it reproduce. There are no sex organs to help it reproduce, it's one cell. So that one cell will become two. The two little cells will become 4 and 8 and 16 and 32, and so we carry on. So that's how viruses grow. Not virus, bacteria. That's how bacteria grow. It's a unicellular organism. Amoeba grows like that. Unicellular organism. One, and then it duplicates itself, and it duplicates itself again, and it duplicates itself, and it carries on. So we go in mitosis, we'll start off with 1. 1 will make 2, 2 will make 4, 4 will make 8, 8 will make 16, and before you know it, you have a complete bacterial infection. All right, now, that's mitosis. Now, how is meiosis different? Meiosis is for sexual reproduction. Okay, now, sexual reproduction means I have to take a half from the male, a half from the female, and I take the two halves and I put them together. Okay, Indy, does it make sense?
Makes complete sense.
If I took a whole male and a whole female, and I put them together, we are going to have some real strange organism. All right. It has to have half the chromosomes in the egg cell and half, or the female cell, half from the male cell. We put the two halves together and we end up with the whole. Now, we are diploid. Diploid means we have a full component of chromosomes. Now, as humans, for example, we have 23 pairs of chromosomes. Now, it's like pairs of socks. If you have 2 feet and you only have one sock because the washing machine ate it again, because there's a little gremlin in the washing machine that steals socks, okay? Well, hello, you don't have a sock for each foot. And you would look rather stupid going out with only one sock on one foot and the other foot without a sock. We need socks in pairs. The same with our chromosomes. They are always in pairs. So even if you get a diagram, people, during meiosis, and you get a diagram that only has one chromosome, you know that they're trying to catch you out. Why? You can never have an uneven number of chromosomes. They are always in pairs. They are always even numbers. So 23 pairs means I have 46 chromosomes. And that is normal for human beings. All right, now, of these 23 pairs of chromosomes, we are going to have 22 that are somatic, which means they form part of body cells, and 1 is going to be your sex chromosomes. All right, but it's a pair. Now, of this pair, one came from your mother and one came from your father. If you happen to be a wolf or an Alsatian or a Doberman Pinscher or a mini Dachshund like my little Bobby Brown, you would have had one set of chromosomes or one set of the socks came from his mom, a sausage dog, and the other set came from his daddy, also a sausage dog. And you put them together and I have my little Bobby Brown. And that's exactly the same as us humans. In fact, any multicellular organism that undergoes meiosis, sexual reproduction. So we have our 23 pairs, this is for humans, and we have, which makes up 46. 22 are for body cells and 1 is for sex cells. These sets are going to be XX for female and XY for males. All right, now, during meiosis, what do we need to do? We have to take the diploid cell, okay, it's diploid, which means it has one set of chromosomes from the mom and one set of chromosomes from the dad. And those ended up coming together to make the baby, which is, or the offspring, which is diploid. Now, during meiosis, and this is how it's different from mitosis for growth, repair, replacement, and reproduction in unicellular organisms, here we have to halve that. So that when I have my half, I'm going to have my whole, and I end up with a half and a half, and during fertilization, I put the two halves together and I end up with a whole again, which is the baby. All right, so how do we do this? We say, okay, easy enough, piece of cake. We have the diploid cell. It is going to undergo the first meiotic division. And what the first meiotic division is, it's called reduction division. Why? Because it takes the 2N and it divides it into an N and an N. So it literally halves the process. It halves the chromosomes in that cell. And it says, you know what? Let's split it. We have to take one set of the pair. It's going to go one side, and the other set of the pair will go that side. And that's what they do here. So it's reduction division, and we call that the first meiotic division. And then our second meiotic division we take this one and it duplicates itself. So this is duplication, and I'm going to write duplication in inverted commas because it's my term for it. Okay, you have a duplication, but your second meiotic division is very similar to mitosis, where what you start off with, you duplicate. So you have an identical copy. And the reason why we do this is that in sperm, during spermatogenesis, which you are going to be doing when you do reproduction, spermato means sperm, genesis means in the beginning, so, or the creation. So spermatogenesis, to make a sperm cell. And this process happens in the testicles, all right? And in the female human, we have oogenesis, because we end up with an egg. All right, so we are making a new egg. We're making a new oogonia. So your oogenesis, we're making an egg. Spermatogenesis, okay, and that happens in the ovaries. And for the male, here we're going to have that process. We're making sperm cells. All right, Now, they are not identical. Why? Are these 4 daughter cells, are they the same as that? No, they're different. All right? Now,
Kathy, why are they called daughter cells? Somebody is asking me why they're not called son cells or boy cells.
you have to understand from a very young age that us girls are more important. Okay?
That is the correct answer.
No, actually, it's the whole thing about mother, father, daughter. All right. So what they did was they speak about paternal being the father, maternal being the mother, and your daughter cell is what results. They could have said your son cell, but then some people could have made the mistake of spelling it with a U, and they thought, well, it's from the sun, and it was just the choice of the scientists at the time. Okay, but they are your daughter cells. And in mitosis, we have 2 identical daughter cells after mitosis, and even Indiana's shaking her head, she understands. And in meiosis, it's sexual reproduction. We're first going to halve or reduce the diploid number, and now we have haploid gametes. So that is haploid. Okay, but now we need to make sure that we have enough. So in the male, those 4 are all going to be sperm, and in a female, one of them will be slightly larger, and that one will become the egg cell, and the other 3 will end up providing nutrition for the egg cell. Okay, so what do we end up with? We end up with this one egg cell being released by the ovary. So this part of meiosis, you have to understand, people, and the reason for it is you need to understand the principles of meiosis so that you can do genetics. And you need to understand genetics so that you can make sense of reproduction. I mean, does that make sense? It sort of follows on. It's a lovely syllabus because you're starting off with where you start from, okay, what you can end up with, and or how the gestation period goes, and what the result could be at the end of it. All right, and then we go on to evolution or diversity, why we are all so different, and we go to genetic variations and
genetic defects.
Yes,
I remember this from last year. It was my best section. I can't wait.
Yeah, and people fiddling with stuff and trying to make things different and artificial selection, all of that is part of one syllabus. So you're starting at one point and you're working your way through, and then you do environment, because it doesn't matter where you come from, where you're going, if you don't look after your environment, I mean, Indy I can see her shaking her head here, if you don't look after your environment, people, well, we're going to have nowhere to go. We're going to just kill each other off anyway, because we're going to poison everything. But before I get on my greens horse,
let's take a break.
Oh, we can take a break.
Guys, let's take a break, and we'll see you back shortly. Make sure that you stay tuned. We've got exciting stuff. Kathy is on a roll, and yeah, see you then. Hi guys, and welcome back. We're having a fantastic session. Kathy is racing through the syllabus. You know what, guys, get chatting to me, ask me lots and lots of questions, because Kathy's going to ask me in a second if any of you guys are paying attention, and that means I'm going to get into trouble if you guys don't post. That's www.facebook.com/learnextra. Also, our Twitter handle is @learnextra. So get chatting and don't get me into trouble. Kathy, over to you.
She makes me sound like a witch. Okay, this is a simplified version of a process of meiosis. I've asked Indy to put this onto Facebook for you, but it's your original diploid cell. You have reduction division, which is meiosis 1. You're going to have your 2 haploid gametes. Then meiosis 2, which is similar to mitosis, you end up with 4 unidentical daughter cells. Then you have your haploid male gamete, which was formed in the sperm as a sperm cell. Your haploid female gamete formed as an egg cell. Fertilization occurs, and guess what? We have our wonderful new little diploid zygote, which has 46 chromosomes. All right, now I'm going to show you a very simplified version of the whole interphase, prophase, metaphase, anaphase, telophase. IPMAT. Interphase is the prep stage. It takes the longest out of everything to happen because it is when that cell, that nucleus, gets all the chemicals ready in order to have the process occur. It's similar to you having a party. It takes 2-3 months to prepare for a really big party, all right, or a wedding, for example, or an engagement party. And geez, when everything's there and the night starts and you have your party, like 5-6 hours later, it's done. But you've been preparing for 3 months. So your interphase is preparing. Then we have prophase. And prophase is when you can always recognize it because in prophase 1, that's your first meiotic division, you are going to have the nucleus, which has now taken over the whole cell because the nuclear membrane is disintegrated and the contents have taken over. And in here, you're going to see your centromeres and your spindles start to form, okay? But the biggest, most important thing is that your chromosomes now lie like this. I'm only going to draw 2, okay? Let's do another one here. And we'll do, those are the ones you got from the mother. And we do the yellow ones are the ones you get from your dad. And the chromosomes are sort of all scattered around, okay, in the beginning of prophase. But halfway through prophase, you end up with this. You'll have the 2 chromatids joined by a centromere, and you'll have that. So let's say this one came from your mom, and this chromosome came from your dad, and they form homologous chromosomes. Homo means the same. Logous means that they're located. So homologous means that they are located in the same place for the same characteristics. So your homologous chromosomes. Homologous chromosomes, one from mom, one from dad. So paternal from daddy, maternal from mommy. Remember, we go on maternity leave, we buy maternity clothing. Maternal is for your mother, paternal is for papa, for daddy. Okay.
Ma and pa.
Ma and pa, right. Okay, so what we've got here is during prophase, this is how you're going to know that it's prophase in meiosis or first meiotic division, is these two start to overlap each other. Okay? And as they overlap, they each pick off a chunk of the other one. So we end up with our chromosomes looking like this. I think I'm being very clever here. Okay. So what have we got here? We've got a piece from the mother's chromosome in you attached there, and a piece from your daddy's chromosome attached there. And this is done randomly. Okay, absolutely randomly. Wherever those chromosomes touch, they sort of do this, and a piece comes from this one into the other one, and a piece from that one goes into this one. So the chromosomes literally swap information so that you end up with this chromosome having a piece of your dad, and this one having a piece from your mom. So you automatically become a mixture. Now it would depend on which one of these two ends up in your baby. So that's why when you look at a child, you can see the child has its granny's hands, and it has its grandpa on his dad's side's nose, and all those different little things. Why? Because you are a conglomeration and a concoction from your mother's, dad's, moms on both sides, and the same on your father's side. So you are all the bits and pieces put together. All right, so this here is called the crossing over. And the point of crossing over here, this point of crossing over is called the chiasma. Okay, it's the chiasma. It's the point of crossing over, but this process is crossing over. So any nucleus, any picture they give you that has that crossing over taking place, you know without a doubt is prophase. Anaphase.
Kathy, I'm so
Yeah.
sorry. The phase, when crossing over takes place, like which phase does that happen?
I said here, prophase. Look.
Prophase.
Yeah.
Okay, sorry, Hudson was asking on Facebook. There you go, Hudson. Hope that
Hudson,
answers your
look.
question.
Prophase, sweet pea, that's when you have your crossing over. All right. So crossing over only in prophase 1, which means it's during the first meiotic division. And the way you write it is you'll write prophase with a 1 here, so that you know it happens in the first meiotic division. Then anaphase is the one I'm going to do next. And anaphase is easy because everything's pulling to, I'm going to take the T off here, everything's pulling to the, if that's the if that's the nucleus that you can see in the cell, you're going to have your spindles. Okay? I'm going to show you an animation just now. So, but everything is now pulling towards, stop. Sorry, it's pulling this way. It's pulling towards the chromosome. Oi. And you'll have one there, and you'll have this one. So they're sort of heading towards the bottom ends. They're pulling away. So that's easy to pick out because prophase, you've got crossing over. And if you can see crossing over happening, you know, meiosis 1. Okay, meiosis 1, prophase 1. Only place it happens. Then if you look at anaphase, those chromosomes are pulling towards at least towards the poles. They're pulling away, that's it. And it's quite easy to see. They're pulling towards the poles. Metaphase is easy because metaphase is always in the middle. So what do you think is going to be happening here? During metaphase, let me just move this down, we have all the chromosomes lined up here on the equator. Okay? All of them. Okay? Lined up on the equator, ready to be zapped off. They're in the middle. They've lined up in the middle. Meta means middle. All right, very easy to remember. Meta means middle. Telophase, easy. And that's our last phase. The T is for telophase. Some textbooks write it telophase and some telophase. So you can either put the E or the O, it depends on if it's American or English, all right? So telophase, you're going to see an invagination occurring here, and then we have what's called cytokinesis, okay? Kinesis means to move, cyto means cytoplasm. So it's telling you the cytoplasm splits. During metaphase, which is in the middle, this divides. You've got one lot of chromosomes going up, one is getting ready to go down, and that tells you that it is karyo. Let's do another color here. It's called karyokinesis, which is the splitting of the nucleus. And you have to know these terms. Karyokinesis, splitting of the nucleus. All right, those chromosomes are dividing. And telophase, we have cytokinesis as it forms the two daughter cells. Remember, this is meiosis 1. All right. Now we go on to meiosis 2. And what I want to do here is show you these diagrams. Okay, so I think it's better than me drawing. Let me just go up here. All you have to remember, prophase, your chromosomes are sitting and they're sort of scattered everywhere, all right? And sometimes they're crossing over and sometimes they're not. So they're all over the show. That's prophase. Metaphase is easy because everything's in the middle. Anaphase, easy because everything's pulling towards the poles. Okay, separating. Telophase, piece of cake, because guess what? It's forming two cells. And if you remember that, to identify the difference between meiosis 1 or meiosis 2, piece of cake. I'll tell you why. In meiosis 1, what are you going to have? During prophase, you're going to see your crossing over. That's number one. Number two, your metaphase, you're going to have double chromosomes. Why? Because we have random assortment. All right, so let's go. Meiosis, cell division, prophase, what have we got? As I said to you, your chromosomes are sort of scattered around. Your spindle fibers are just forming. They're your centrosomes sitting over here. Everything's happy, everything's comfortable. Let me just get a nice pink so we can see. And your chromosomes are crossing over, and that's your chiasmata. As they cross over, they're swapping information, which is very important. All right, next slide. Okay, meta, middle. You see your chromosomes are lined up in the middle, and they line up independently. All right, so what I'm going to do now is I'm going to take, we're going to do this. This was a chromosome that this organism had from its mother. Okay, it's maternal chromosome. These are the homologous chromosomes. They're the pair for certain characteristics. So these two here would be for one lot of characteristics, and I've just moved this down, and I've moved it down horribly. Well, how did I do that?
Oh no, there's...
Here we go.
It's just very sensitive.
I must have pushed something by accident. Sorry, people. Okay, so that's your homologous chromosomes. They're homologous. They are the same for the same characteristics, okay? The genes for the same characteristics are situated there. All right, so that's from mom, and then dad's ones we'll do yellow. They're here. Okay, let's try green. Maybe that's better. Can we see? All right, so what have we got? And I left a piece of mom off there. So that's after the chiasma has taken place, the crossing over has taken place. We've randomly swapped genetic information from dad and from mom and put it together, all right? Now those homologous chromosomes pull away. There's no division of the centromeres. You see them, that's very important. And we have everything lined up at the equator. It is in the middle. All right, next one, anaphase. Here the chromosomes have pulled away, and they have randomly pulled away. So we could have had dad's one pulling, dad's one here, mom's one there, and mom's one here. But we could also have had it the other way around. So if I have 4 chromosomes, 1 and 2 are a pair, and 3 and 4 are a pair. 1 and 4 can end up being on one end and 2 and 3 on the other end, or it can be 1 and 3 and 2 and 4, whatever the case is. It is random assortment. It's independent. There's nobody knows how it's going to pull out. That's why you do not look like your mother's 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 mother for the last 10 generations. You look different. Why? You look like a combination of all your family traits. Why? Because of this. 2 things. 1, the chiasma. 2, the random and independent assortment here. The chromosomes go randomly. All right, next one. Now we go meiotic. The first meiotic division, there's your telophase, and we have a double-stranded chromosome. Cell membrane starts to constrict. So we have cytokinesis. The cytoplasm has now divided completely, and eventually we end up with the 2 daughter chromosomes. But people, they are not identical. Why? Because The one's got more pieces from the mom, and the other one's got more pieces from the dad, and you've got dad's one's up here and mom's one's up there, maybe 10 dad and 2 mom and 5 moms and 3 dad, whatever the case is. Independent assortment, all right? And that's what we end up with. 2 unidentical daughter cells. Now we move on to the second meiotic division. Now remember, here we are duplicating. So we're taking our haploid gamete and we are now making an identical copy of it. All right, here, and that's why in your diagrams you will see that they actually put the 2 diagrams there, and that's another indicator that we are dealing. I didn't touch this thing. You know, I'm quite sure that this screen just does what it wants to do to me. Okay, I need to go back. Okay, I've lost it.
It's okay. You find it, and I'll chat to the mindsetters. I'll have Dudu by my side. Guys, thank you for sending in all of your questions. I just wanted to let you know that you can help each other out on the page. That's what we love the most, is when there are mindsetters that are struggling, and instead of waiting for us to answer it, you go and you start answering those questions and helping others out. Guys, don't forget that we have Life Science every Wednesday, and we also have Learn Extra Live every week, Monday to Thursday. Grade 10 starting at 4, grade 11 starting at 5, grade 12 starting at 6. And let's see, Kathy, how's it going?
I've got it. I don't know where that page disappeared to, but here we go. All right. Now, in the second meiotic division, we have metaphase. It's middle. All your chromosomes will be lined up in the middle. But people, please look at this. First of all, I've got 2 nuclei that they're showing me. But instead of having 2 rows of chromosomes, how many have I got here? I've got 1. See? 1 row. Whereas in first meiotic division, in other words, meiosis 1, I had double. I had my homologous chromosome sitting there. Now, I've only got 1 row of chromosomes. Okay, now what happens is this centromere splits. And the minute that centromere divides, so that the daughter chromatids can move towards the poles, we say karyokinesis has occurred. Why? Because karyokinesis is the splitting of the nucleus. Okay? There's our anaphase. Anaphase, very easy to identify because the chromosomes are going to the poles. And in anaphase 1, our chromosomes had little butterflies like this. Okay? Pulling across, and here they look like that. And the reason I'm telling you this is that I'm not sticking to all the academic biff, is because I need for you to understand, to be able to recognize what you're looking at. Okay? So your daughter chromatids, not your son chromatids, your daughter chromatids, are pulled towards the poles. As soon as they get to the poles, cytokinesis takes place, and they are then divided into 4. But are they identical? No, because in mitosis you end up with 2 identical daughter cells with the same ploidy. Here they are half. So these are all haploid structures. They're haploid, all right, and they are not identical because they all contain bits and pieces from the homologous chromosomes that split in meiosis 1. Are there any questions, Indy?
We do have a few.
Okay, ask.
Okay, are you ready? Let me have a look here quickly. Let me just have a look. Sorry. There's a few people that are saying, guys, what I need to know is when you're not understanding, instead of saying, I don't understand what's going on in that section, rather say, I don't understand what's going on in that section. Here's a bit what it's about, because it's much easier for us to help you. I'm just trying to find a good one for you. I keep asking the mindsetters to send in a really, really hard one for Kathy. Guys, we've got to get her. Abu wants to know, why is meiosis essential? See, guys? See?
Okay. Guys,
I knew. Guys.
Okay.
Guys.
Look, if, okay, Abu, hopefully at some point in your life you're going to meet somebody that you absolutely love. And maybe with a bit of luck it happened yesterday during Valentine's Day, okay? But you get married, you do your thing, whatever the case is, you decide, you and your partner, to have a baby. Okay. Now, your partner, as she sits there, has 46 chromosomes. Okay? In other words, she is diploid. All right? That's what you're going to find in every cell in her body. Okay? This. Now, in your body, look at this. And this is something that all learners tend to battle with. And that's why I've gone about it the way I have. Okay? So this is normal. Okay? This is what you're going to find in any cell in your body and in her body. Your somatic cells. All right. Now, in the sex cells, or your sex chromosomes, you are going to have X, Y, because that Y is what makes you male. And the girl of your dreams is XX, because that's what makes us girls so special. See, you guys are missing a piece there. We aren't. We're perfect. XX. Wink, wink, nudge, nudge, say no more. And Indy's even having a good smile. All right, now, watch here. Okay? The two of you want to have, what's the boy's name?
It was Abu.
Abu. The two of you now want to have a child. Now, if there's no meiosis, so let's take a different color here. If there is no meiosis, we can clone, all right? Cloning would be to take any one of your somatic cells and take the nucleus and replace the nucleus in an egg cell and take that egg cell and stick it in a person and make it grow, which is completely, totally, 100% unethical, and anybody found doing it, whoa, they would be in deep, deep trouble. The best they've done is Dolly the sheep, where they've cloned. So the normal process is not cloning. The normal process is sexual reproduction. But now look at this. There's no meiosis. I'm going to have 2N from there, 2N from there, and guess what I'm going to end up with is 4N. That's polyploidy. We're going to have a problem here. Can you imagine a human being with 4N? Okay, so it's got instead of 2 lots of chromosomes, it's now got 4 lots of chromosomes. This is going to be, I don't even think that your loved one, your spouse, is going to have to even be able to carry that in her womb. All right, so what do we need? We want another 2N. Don't you want a baby that's like you? You want a 2N. So how on earth do we do this? Well, guess what? She's 2N, you are 2N. What do we need to do? We need to say, okay, in her reproductive structures, like her ovary, she's going to take this 2N and she's going to turn it into N. And that's going to make the egg. And in your body, you are going to take, in your testicles, you are going to take that 2N and you are going to change that 2N into N. All right? And that's going to be your sperm cell. Because guess what? It only takes one little sperm to get there on time and fertilize that egg. Okay? So, then I take the sperm cell and the egg cell, and we have fertilization. And I have N plus N. N plus N. And what do I get? Well, easy. N plus N is 2N. So there, this little baby is now perfect, okay? And it's got one set of chromosomes from its mom. It's got one set of chromosomes from the dad, and you put them together, and that's that. But now you must remember, this set of chromosomes it gets from its dad, in there, it's going to have some chromosomes from his dad. It's going to have some chromosomes from his mom, all right? It's going to have mixed chromosomes from his mom and his dad, where we had that kind of thing, and, oopsie, that one with, all right. So what's in this sperm cell, the chromosomes in that sperm cell, the genetic makeup in that sperm cell is a combination of you, Abu, and your mom. I mean, you, because you are a combination of your mom and your dad. On this side, we've got the same. She's going to have exactly the same spread, where she's going to have some bits and pieces from her mom, some bits and pieces from her dad, a whole chromosome from her mom, a whole chromosome from her dad. But here, there are 23 chromosomes. And here, there are 23 sitting in there. And they go together to form this perfect little baby. And that baby has got your genes and your mom's genes and your dad's genes and your grandpa and your grandma's, and the same from her side. So this baby ends up being you. Why do we have meiosis? Because we have to take the two diploid structures and convert them to haploid structures. So when we take the diploids, we turn them into haploid, and we then make a new baby. Because you can only forge a baby together with two gametes that are haploid, not a diploid cell. All right, so that's why we do it. Any other questions?
There's quite an interesting one here, Kathy. It's from Tumelo, and Mbadi's actually commented on it, and she said that she actually got asked it in a test this week. I want an example where duplication of chromosome numbers does not lead to a genetic disorder or death.
She wants an example of where a duplication
of chromosomes numbers does not lead to a genetic mitosis.
Remember, mitosis is growth, repair, replacement, and reproduction in unicellular organisms. People, they are quite capable of asking you a comparison, piece of cake, draw a comparison or a table between meiosis and mitosis, all right? But they could also ask you a trick question like that and say to you, okay, well, what kind of cell division takes place where it doesn't lead to any chromosomal malfunctions or mess-ups or anything? It's mitosis, because meiosis hasn't occurred. You're not halving the chromosome number. Now, what I really would like to do is show you this, okay?
Oh, this is awesome.
What I'd like you to actually go and have a look at this. It's at Cells Alive, so plural of cells, cellsalive.com, and go and have a look at it.
I will post it right now, guys.
Please, because it's really nice to watch. But here, I'm not going to play it because it plays through too quickly, but I'm going to go step by step. So we've got this diploid cell. It's 2N. It's got a set of chromosomes from its mom and a set of chromosomes from its dad. So that's what we've got here. So there's prophase. What have we got? Well, the nuclear membrane disappears, it disintegrates. All right, so I'm going to just draw, I can't. You have the nuclear membrane disintegrates, the centrosomes, because remember it's an animal cell that we're looking at here. In a plant cell, you won't have any centrosomes. But here you've got the centrosomes, and the centrioles, and what do we end up with? The spindles developing, and they're your chromosomes. They've got a red and a blue, and a purple and a yellow. That's prophase, all right? But can you see here how this blue is sort of sneaking its arm across? And I'm going to wait for my producer to actually come in onto the screen so that they can actually see, so you can actually see it. All right, it's almost like this blue one's got its arm around the red one here. Okay.
It does.
All right. I've got the little arrow. If you can see the little arrow moving on the screen. Okay. That's prophase. We have a chiasma occurring. It's the crossing over. It's the swapping of genetic material. Now, metaphase, everything is lined up in the middle. But look here. We've got a piece of the mommy chromosome sitting on the dad. We've got a piece of the dad's chromosome sitting on the mom. We've got the same here. So a piece of red on the blue, and a blue on the red, and the yellow on the purple, and purple on the yellow. So that is after your chiasma. But why? How do we know this? Metaphase, they all lined up in the middle here. You see? They're in the middle. All right. Anaphase, they start pulling, but they pull randomly. So what we could have had is a red one, we got a red one, a blue one with red and a purple with yellow. Here we've got yellow with purple and red with blue. They could have pulled the other way. We could have had this one going there and that one coming there. And we could have had it the other way around here between those two. So it's what we call independent assortment. It is independent of anything. And it's because of this independent assortment that we have genetic variation. So it's the chiasma, okay, and the crossing over in prophase 1 gives us genetic variation, and this independent assortment of the chromosomes. Nobody knows which one's going to go in which direction. Okay, then we have telophase. How do we know this? We have cytokinesis occurring. Here, you see, we have, if you can see this little arrow that I'm using here, that's what we have there, and we have this invagination develops, and our little chromosomes are sitting way at the end, and we have a division that happens in the middle. So what we've got here now is two unidentical daughter cells. Why? This one is red with blue. This chromosome is yellow with a touch of purple. This one down here is a purple with a touch of yellow, and this one here is blue with a touch of red. Are they identical? No, they are unidentical daughter cells at the end of meiosis 1. Now we go to meiosis 2. There we have prophase, and very similar to the other one, except there's no crossing over. Metaphase, everything is in the middle, but look at what we've got here. We don't have double chromosomes lining up here. It's one chromosome on either side. And also, something else. Meiosis 1, that centromere doesn't divide. In meiosis 2, the centromere divides. So, metaphase, everything lined up. Anaphase 2, they start to pull to the poles. But here again, it's independent assortment, because this one here, this red one, if you look here, that red one doesn't have any blue. This one is red and blue. This one could just as well have gone to the left, and that one could have gone to the right. So we have independent assortment again. And then telophase, they separate, and what do we end up? We end up with our 4 unidentical daughter cells. And in the male, this happens during spermatogenesis in the testicles, and in the female, it will happen during oogenesis in the ovaries. If it's a plant, well, in the anther, you know, you've got the stamen and the anther. The anther, where the plant makes pollen grains, you're going to have spermatogenesis occurring there. And in the female, it will happen in the ovary. All right. So what have we got happening? Whether it's the male, whether it's a plant or an animal, if it's a male, it's got certain reproductive structures. If it's a female, it's got ovaries. Okay. But what do we do now? We say in the male, all 4 will become sperm cells. But 2 of them will be X chromosomes and 2 will be Y. Because remember, a male is XY. And when we have reduction division, one gamete has X and one has Y. And they duplicate, so I've got 2 X's and 2 Y's. And what makes a male is when an X sperm cell bonds with an egg cell. So X, Y. And if an X sperm cell bonds with the female, then it's XX, and you end up with a little girl. And that, people, please just go and have a look at this. It's so nice. You can actually watch it all moving, and it happens quite quickly. So, and you can also stop it. You can have a look. Just remember all the little indicators that I told you, because what they're liable to do is give you a line diagram and say, identify. What phase is this? What is happening? Why is it happening? Over to you, Indy.
Thank you. Fantastic, Kathy, for an amazing 3 hours of life science. Grade 12s, you've been fantastic. Thank you so much for asking so many amazing questions on the Facebook page. Don't forget, guys, tomorrow we've got maths lit on. So please join us, grade 10s, grade 11s, and grade 12s. That's on tomorrow. And also, keep checking back on our Facebook page. And thank you for an amazing session. Also, before we go, Maricela, thank you so much for your amazing post. She said that she wants other mindsetters to help each other out. So guys, once we're gone, do just that. Help each other out. Mindset learn more, learn extra. Bye guys.